Welcome everyone to this Red Talk featuring Professor Jude Fitzgibbon. I truly feel honored having been invited as a co-moderator together with Dr. Sandrine Roulon. I have to disclose that I'm a first-time participant, so let me quickly introduce myself. My name is Oliver Weigert. I'm a physician-scientist in Munich, Germany. I work as a hematologist-oncologist at the University Hospital. I hold a professorship position for translational lymphoma research at the Ludwig-Maximilians University, and I'm a member of the board of the German Lymphoma Alliance. Sandrine, do you quickly want to introduce yourself? Probably no need to, but...
I will. Thanks, Oliver. So it's also my pleasure to moderate this session with you, Oliver, and to host Jude for this Red Talk. So my name is Sandrine Roulon. I am a research director at INSERM, and I'm also group leader at the Center of Immunology of Marseille Mini, where I lead a team working on the B-cell pathogenesis and targeted therapy. So, Jude, I think.
Thank you.
And
so maybe
Oliver for the introduction.
Exactly. I was asked to quickly introduce Jude, probably also no need to do that, but it's a pleasure to do so. Probably very well known to many of you for his seminal work actually on genetics and epigenetics, both in familial forms of AML, MDS, but of course also malignant lymphoma, I guess follicular lymphoma in particular. As you know, he contributed significantly to our understanding of clonal evolution and essentially established the concept of common precursor cells, which we will talk about today. He also spearheaded the field of aberrant mTOR signaling, as well as targeting of KMT5 and KMT2D mutant lymphomas. But beyond his scientific excellence, to me, Jude has meanwhile become not only a mentor, but a long-term collaborator and friend. He did his bachelor in genetics at Trinity College in Dublin and received his PhD at the University College in London. Not without some regret, at least from an academic point of view, I have to say that he recently has taken up a new appointment as Vice President of Haematology Discovery and Target Biology at AstraZeneca. Previously, he was a professor of personalized cancer medicine at Barts Cancer Institute in London, and he fortunately retains his honorary appointment there. He has an impressive academic track record, held numerous leadership positions, including, but not limited to, the chair of the UK's National Cancer Research Institute Lymphoma Science Subgroup, and of course, he's an associate editor of the British Journal of Haematology, just to mention a few. And with that, Jude, the stage is yours, and we are very much looking forward to your talk.
Excellent. Really, it's a fantastic pleasure to be invited to such a prestigious meeting, and it's even wonderful to have Sandrine and Oliver introduce me, because I know them very well. I'm going to talk a little bit about the work that they've done, and hopefully over my 45 minutes or so, just tease you a little bit in relationship to where I think the field should go and is going, and hopefully that we can cure the incurable. Just want to see, can my slides pop up? Oh, excellent. And we can see whether they can move on. So first of all, I'm going to talk about targeting the CPC population, follicular lymphoma, but it's a little bit of a cheat in relationship to a title, because I'm really only going to touch on it at the end and really talk a little bit more about the journey in relationship to getting to that CPC population. And I can imagine we've got a kind of a breadth of different researchers online today, you know, some deep thinking, very immersed in the field, and others maybe pretty new to lymphoma. And so I'm going to try and explain my academic journey a little bit as well, and how your thought process evolves. And I'm talking of somebody who, when they started their PhD, to be doing PCR by water bath. And when you think of the complexities of single cell analysis, computational tools at the present time, the opportunity to move from a bow and arrow type research program to something that has a lot more qualities and depth to it, I think is very exciting as we try and understand these diseases and generate cures. As Oliver has said, I'm now a VP in Haematology Discovery at AstraZeneca, but I've just started a few months. So everything that I'm going to talk about today really is about my work in the Barts Cancer Institute. So here's the second slide. So I spent about 25 years at Barts, and I think what everybody here needs to understand is it's all about getting the question right, because there's always going to be a certain amount of luck, and there's always going to be a certain amount of talking and meeting the right people at the right time. But invariably, if your work isn't of sufficient quality or you're not thinking deeply enough, it's going to be very difficult to be successful in research, because there's a lot of high quality individuals. Also, I think you have to be lucky by identifying or seeing something early that gives you a sense of the essence of a disease. And my clinical understanding of the disease, and I think what really has informed my research, is an example here of a bone marrow transplant or donor-derived lymphoma. So Dad was a bone marrow donor for his son, and both developed follicular lymphoma 3 and 11 years post-transplant. And immediately that gives you a sense that there must have been some cell that was transferred at the time of the bone marrow transplant and gave rise to follicular lymphoma in both donor and recipient. And it brought home to us, and this is now going back 12 and 13 years, the notion that evolution is going to be important. And that's not necessarily surprising. So that you have your follicular lymphoma, you have your 14/18 translocation that's synonymous with the disease for a long period of time. And you get an evolution in relationship to these 14/18 positive cells. What I'd say to everybody is, as I go along, if anybody has any questions, please pop them into the chat, and I can introduce them into the talk itself. And so this is the fourth slide, and just really to give you a sense of the disease. You know, we could say it's easy to say it's incurable, but some patients stay in remission for a long period of time. It clearly has a relapsing remitting clinical course, and this is quite an extraordinary patient that was diagnosed in 2006, and you know, we followed the patient's treatment. We've taken biopsies at different stages. We've done gene expression and whole exome sequencing to try and understand the genetics as the patient evolves. But it really shows that relapsing remitting nature of the disease, and you know, the difficulty encountered in therapies work, and it's an enigma of sorts that the therapies work well, but actually patients continue to relapse. And I think one of the early mentors that I had was Professor Andrew Lister, a clinician, you know, and he had identified these questions. How can you actually explain something like this happening? Also, I think when you look at survival curves from patients, and you know, this is one of the most recent articles looking at a large number of patients, that you're not getting a plateau in the curve, again suggesting that patients continue to relapse late and succumb to what is an indolent disease, because about 80% of patients are alive at 10 years. And this is just a schematic that somebody created for me, you know, a number of years ago. But what I like the notion of here is to look here and think about patients with disease, and then episodes of overt symptoms, overt disease, and then minimal residual disease. And I think that's something that's important, because those minimal residual cells are probably going to be important in how we try and identify what's causing and how can we better target the occurrence of relapse in these patients. I think it'd be fair to say that predicting outcome in follicular lymphoma is tricky. And that's because patients can live a long time with the disease. So predicting outcome at diagnosis is tricky. Improving overall survival has proven very challenging. Outcome is good. And understanding one of the key features that actually drive poor outcome is histological transformation. And you know, that occurs probably now with the introduction of our chemotherapy, probably maybe 5 to 10%. And there was a nice paper by the French group in JCO, which I use a lot when I'm thinking about the disease. But you have to think about follicular lymphoma, and think in relationship to histological transformation probably being responsible for the majority or for a similar amount of deaths compared to frank follicular lymphoma itself. And also, I think Oliver and several others have introduced the notion of POD24 as a kind of a sense, just to give us a sense of which of the patients that are doing badly, why are they doing badly, and clearly this is an area of a clinical need. So when you look at the challenges, I think what I want to get across to everybody is, we're not looking across the board. Yes, from a genetic point of view, it has a 14/18 translocation. And you'll see later that some of the mutations occur at a very high frequency. But there's a lot of clinical heterogeneity here. And I think that generates a lot of challenges, because when you're trying to take an individual who's presented with follicular lymphoma diagnosis and say, can we predict the likely outcome subsequently? We have to keep in mind that 20% of patients on watch and wait will never require treatment. Yes, you've got patients that are likely to relapse within 24 months. About half of the deaths will occur by transformation, and transformation doesn't necessarily have to be the first event. It could be later in the evolution of the disease. You also have got 14/18 positive and negative cases. And you also have to keep in mind the heterogeneity in relationship to the biopsies, because if you take a biopsy and you use that as your basis on which you collect mutational analysis or gene expression profiling, all it's providing you is the notion of what's happening at that site. It could very well be that something very special or something very important might be occurring at other sites where there are tumor cells, but you won't actually be able to capture that information. And so I think that people start on the basis that you want to define outcome based on a diagnostic follicular lymphoma sample without necessarily factoring in that there's a lot of heterogeneity here to capture. And you're not looking at outcome effects based on a single event like FL only or transformation only. And there's lots of different things to factor in. And I think that's why all these studies, including really nice work by Sarah Hewitt and Leo, looking at gene expression data, Oliver's pivotal data in relationship to the M7 Flippy, and several others. And what all these things have done is they've set the bar. They've set a bar for what kind of test is actually going to be sufficiently informative for a clinician to decide, yes, we will now change our treatment at diagnosis because this patient is of higher risk. And I don't think we're ever going to be there, because I think it's too high a bar given the heterogeneity. But that's open to discussion. And I want to focus a little bit on transformation here as well, because I don't think we still have a handle on what causes transformation. I think, yes, there's lots of different reviews, and we've all written them and we've all read them, but in a sense, it either is a multitude of different factors that are driving histological transformation. We haven't really the right microscope yet to understand what's the cause of transformation itself. And this was a nice paper in British Journal of Haematology that looked at transformation and then broke it down into when transformation actually occurred. Was it during a period of watch and wait? Was it first relapse? Was it a later relapse? And then the notion of composite and discordant, which is slightly different to what we'd call transformation. But what we tend to do then when we're actually doing experiments is we lump all these in circumstances together. So they're conveniently lumped together because it's very hard to get sufficient numbers of biopsies to actually ask discrete questions about what's happening at each individual stage. So you can imagine, if you transform to high-grade disease on watch and wait, well, that's quite interesting. It's quite a clean system potentially to be looking at histological transformation. While if you're looking at it later, or you're making comparisons with a follicular lymphoma biopsy that might be in a different location to the biopsy that's taken for histological transformation, that's something quite different. I think the other heterogeneity component I think is important to keep in mind is based on gene expression data. We now know that patients can transform primarily to GCB type diffuse large B-cell lymphoma, You also have a small number, but significant number of cases that are ABC derived. And there's a new articles by the Vancouver group, which show there's a mediastinal group of transformed cases as well. Again, heterogeneity. And we're lumping all these together, looking for clues that might explain what's driving the disease. And maybe that's why we don't have enough cases of the right type, or we're not dividing patients into groups and then asking discrete questions to get a definitive answer. Now, this is the genetic landscape of follicular lymphoma. And I'll first highlight on the right-hand side kind of work we did, 7 or 8 or 9, 10 years ago, whereby we had sequential biopsies from patients, and that allowed us to actually trace the evolution of the disease over time. And this is standard now, but at the time it was excellent to have biopsies taken at different stages of disease, but also then to be able to use next generation sequencing. What is next generation? At the time, either whole exome or whole genome sequencing, where the tools available were really just being developed. And I think that gave us a sense of the evolution of the disease. I think that what's on the side is this is work really done and driven by Oliver. And I use it because I think it's a really excellent schematic showing the presence of mutations in follicular lymphoma in about 430 cases. And what you can see immediately when you look at other cancers is you can see the frequency of mutations. Mutations in KMT2D, CREB-BP, BCL2, several different genes. And so many patients have, to keep in mind, many patients have multiple genes, mutations in epigenetic regulators. And secondly, the frequency of these mutations are very high. So you'd say that the core genetics of every follicular lymphoma patient with a 14/18 translocation and multiple epigenetic regulators is quite distinctive. And I think this is highlighted here, whereby in a way you can see that all the mutations that are shown in yellow are all chromatin regulators. And so there's something special about follicular lymphoma that is reorchestrating a gene expression in some way within the tumor cell. And that allows us then to think of different questions that could be important. But I think KMT2D and CREB-BP and EZH2 are 3 key ones. And we can talk a little bit about them as we go along. But again, this work has been contributed to by several different groups around the world. And today I'm going to focus on the work that we've done primarily, dipping in and out a little bit. But more so, I think, just to give people a kind of a sense of the direction of travel. And so you could say that in addition to the 14/18 translocation, the second genetic hallmark of follicular lymphoma is epigenetic mutations, where you've got a histone metal transferase called KMT2D that creates an activatory chromatin mark, lysine 4 trimethylation is mutated, loss of function. You've got CREB-BP mutations, a histone acetyl transferase, and you've got EZH2 mutations that create the lysine 27 trimethylation mark. This is a gain of function, so you get increase in this activation mark. And across the board, you seem to be getting increase in these inhibitory factors. And I think that this leads to another key question that I don't think has been addressed. But what people have done very nicely is that they've looked at the biology of each of these mutations, why they're important, what they might be doing, not just to the tumor cell, but also its interaction with the tumor microenvironment. Lots of different papers. But I think what hasn't been addressed is all these individual genes have individual functions, or are they additive in some way? What exactly are they doing together? Because a lot of patients will have multiple different mutations in epigenetic regulators. And I think that what's happened thus far really is that people have dipped in, they've used a genetic background like BCL2, like MYC overexpression, they've added in one mutation and asked the question, what function has that in relationship to lymphomagenesis, etc. And I think the question really is what are the additive effects, or is it a multitude of separate effects that each of these individual genes do? And I think it seems sensible to suggest that there must be some overlap. And when you look at the gene expression changes that accompany certain mutations, there clearly is. And can we tease apart, is it an overall malaise in relationship to gene expression, or is there specific nodes that are important? And I still don't think that those have been answered. And these things take time. So, for example, Ryan Moran was the first to report mutations in the histone methyltransferase EZH2, and our group went on to show that these were highly frequent in follicular lymphoma. And since then, there's been, a plethora of work looking at EZH2 inhibition, an example of precision medicine in follicular lymphoma, and the development of clinical trials in this area. I suppose the work that we did was targeting or thinking about the most frequent gene mutation. And the frequent gene mutation is in KMT2D. It's a histone methyltransferase. It's also mutated in a syndrome called Kabuki syndrome. And we reasoned, and this was really a very nice commentary that was on our paper in Blood. And I'm going to walk you through it, because we reasoned that if you lose mutations in histone methyltransferase, and because it's loss of function mutations, how can you actually ameliorate the effects of that mutation? And so we reasoned that if you inhibited the corresponding demethylase, that essentially is removing the mark, you might be in a position where you can exert effect or revert the effects of a specific mutation. In this case, a mutation that not only is present in follicular lymphoma, but it's also present in the GCB type diffuse large B-cell cases, and a smattering of probably a lot of hematological malignancies, but also a lot of other solid cancers. And I just thought this schematic was really nice. So in the normal setting, KMT2D, in its methyltransferase role, adds on these little red trimethylation marks, and the demethylase, and I'm using my arrow, but you won't see it, but I'm using the demethylase essentially removes those marks. So you've got a balance between creating an activation mark and removing it. In the malignant scenario, and because a lot of the KMT2D mutations can occur they can occur in the heterozygous state, but also they can be compound heterozygous, so basically biallelic inactivation. What you can see here is that you're losing that opportunity to create these marks, and then the methyltransferase has activity, and so therefore will remove those. And therapeutically, you can see that if you can inhibit in some way the demethylase, you can maintain hopefully enough or sufficient residual trimethylation to result in activation of genes. And so what we showed here is that if you use different inhibitors, you can actually see that the chromatin mark in this Western blot without treatment and treatment actually increases. So in a way, you are reverting the effects of the mutation. And we could show that in some examples, we could get a sense that we were reducing BCL2 levels, even though we don't think that that's the key factor. We don't really know what the key factor is that's driving the effect that we're seeing in vitro, whereby we're seeing that if you have mutations in KMT2D in lymphoma cells, they're more likely to die. Some modest in vivo activity as well. But all in all, kind of giving a sense that we can think about these specific mutations that occur at a high frequency and see can we actually revert and change them. I think this leads to another question. What's important? The catalytic or the non-catalytic activity, or both? Because what you can see when you start delving into epigenetics, you can see that these proteins are all interrelated. They're pulled together into various complexes. So that targeting the methyltransferase activity or the acetyltransferase activity is one component, but also the targeting themselves can be important because they're all interrelated with each other. And one example I'm going to show here is that KM2TD is mutated in 90% of these Kabuki syndrome patients I explained. It's a developmental disorder. 10% of patients have mutations in KDM6A. That's another gene for UTX. And so it also shows the promiscuity in relationship to epigenetics is that one is affecting KMT2D2C is affecting the lysine 4 mark, while KDM6A is the demethylase affecting the inhibitory chromatin mark 27 acetylation. So you can see, you could ask the question, well, is it the epigenetic, is it the catalytic function, or is it the non-catalytic function, or is it both? And then you can ask the question, are the effects affecting the tumor cell, or are they having a knock-on effect instead of on the tumor microenvironment? And so you can see how you can building up a really complex picture of the potential interrelation. It's like a soap opera. It's really trying to tease apart those associations and to weigh them up so that you get a sense of what's important and what's not. And I like ideas, and there was a really nice idea proposed in a review article, and so you can see that KDM5 or KM2TD, the histone metal transferase, creates this activation mark, histone H3 lysine 4 trimethylation. And then you can see that depending on whether you have a mutation, you can start losing this particular mark, or the mark can change in some way. And so you can see here, let's say in the wild type setting, you create a kind of a very homogeneous population of the mark is laid down on the genome in a very robust way, which by definition then creates a very homogeneous gene expression signature. While if you lose the mark, potentially that might affect the size of the mark, the breadth of the mark. And in some cases, some cells will behave like wild type, but other cells will start having a more heterogeneity in relationship to how the mark is expressed across the genome. And so therefore, within an individual cell, certain genes might have a perfect chromatin landscape, while others might have less so. And that creates heterogeneity. And I think what we're beginning to see is that heterogeneity is key in relationship to evading treatment and providing cells with an opportunity to become resistant potentially to disease or to treatment and then subsequent relapse. But I like this idea of thinking in relationship to the heterogeneity that these epigenetic mutations might be creating, because they're affecting so many genes and you're not actually generating a perfect chromatin landscape around an individual locus, and by definition then keeping a very tight control of gene expression. But it's an idea, nothing more. So what other genetic mechanisms are there that are interesting about follicular lymphoma that make follicular lymphoma unique? Well, as Oliver said, ourselves and other people, I really have to shout out to a colleague, an ex-colleague of mine, Dr. Jessica Ockeson, who basically was looking at our data, but also looking at gene expression data or mutational data from other different centers. And she spotted before anybody else that maybe in the supplementary data, people had been missing out on a type of mutation. And this mutation is in the mTOR pathway. And key here is that these mutations in RAGGC and ATPases lead to subtle but measurable changes in mTOR activation. But at least in relationship to RAGGC, they occur in maybe 10 to 15% of follicular lymphomas, but they're unique to follicular lymphoma. And it's that uniqueness allows you to ask another question, well, why are they unique? And I don't think we've actually been able to get an answer to that. What we have been able to do in conjunction with really a fantastic group in the CNIO in Spain, Alejo Efian, and I want to give a shout out to Ana Ortega Molina as well, who worked previously with with Guido Wendell in Memorial Sloan Kettering, and published one of the two seminal papers on KMT2D. Here, she's actually looked specifically at the effects of RAG-GC mutations and shown that they can drive follicular lymphoma. But why specifically follicular lymphoma? Why not other forms? I think is an open question. And maybe there's some clue here as to the special features of follicular lymphoma. So what I'm trying to kind of, create a picture to start with. What I'm trying to create a picture to start with is the complexity that we have here, and that we've tried lots of things. We've generated lots of data. We know a lot about the disease, but we still haven't quite grasped the way of is there a way of trying to cure follicular lymphoma going forward? And so I just want to show three examples of work, and one dates back to the 2002, where we, along with several other groups who had done this previously, but we used real-time quantitative PCR, nothing to get excited about, to show that in a large population of healthy donors, about a quarter had the 14/18 translocation. So this is going back now over 20 years. And you kind of wonder then, well, how important is the 14/18 translocation? Because a quarter of the population are carrying around these rare populations of cells. As I said, we have these two examples now, and one of them, look, Oliver's work when he was in America at the Dana-Farber Cancer Institute, and ourselves, two examples of donor-derived follicular lymphoma after stem cell transplantation. Again, giving us the notion that there's these 14/18 positive cells that have experienced the germinal center, that have undergone somatic hypermutation, have acquired some mutations, but and can lay dormant for long periods of time before they change in some fashion, and lead to overt disease occurring. And then we've got longitudinal profilings that I showed earlier on, whereby clonally different biopsies from the same patients are related to each other, of course, from a genetic point of view. But if you draw a phylogenetic tree, you can see that they're potentially more related to this so-called progenitor population called the CPC. And I think what we haven't been able to do is to put our fingers in, to grab that cell and say, what do you look like? What are you saying to us? Who are you? And how can we remove you? But I think there's a tacit agreement that it exists. And the question now is, and I think it's a mini race of sorts, who can actually pinpoint the phenotype of the cell? And it's probably not, as we now know from single cell analysis, it's never going to be a single cell or a single population. It's going to be a kind of a continuum, and that these cells potentially are what might explain, at least what I believe it explains, that relapsing remitting nature of the disease, whereby these cells evade treatment. They might be controlled a little bit like by maintenance rituximab, but they evade treatment and invariably then repopulate new episodes of disease. And I think a shout out has to go to Sandrine and Bertrand for their work. And they've a multitude of different papers, but I just want to highlight this one. It's a really clever idea that they had to say, let's use the epic normal population of patients. Go back and see how many of these subsequently developed follicular lymphoma, pick those out, and ask the question, is there follicular lymphoma biopsy related to the 14/18 translocation that's present in pre-diagnosis of the disease? And they were able to show that it was. And maybe it's not a surprise, and maybe it's what you'd expect, but to have such beautiful experimental data showing you that the 14/18 positive cell went on subsequently to gain some additional mutations, maybe these epigenetic mutations, to drive the disease, I think is really nice indeed, because it cements this notion of a long-lived population of B cells that have experienced the germinal center, that are circulating, and it provides us with a focus on how to cure a disease like follicular lymphoma. I just want to show some more examples of this kind of heterogeneity as well, because what we have here, and this is more work done by the BARTS group led by Jessica and a really wonderful clinician scientist called Shamsa Araf. whereby we had opportunity to look at multiple biopsies from the same patient at the same time. And so there was 22 kind of synchronously removed, spatially separated biopsies. And then if you look genetically at those, you can see that there's heterogeneity across the different biopsies. Again, suggesting that clonal evolution is occurring. And two, that looking at one specific biopsy might necessarily give you a sense of the disease. And I think going forward, I think the introduction of cell-free DNA might in some way be able to get over that, because you can then, the cell-free DNA component is flooding in genetic data from a number of different sites rather than the individual site that you identified in the first place. And maybe this isn't just something that happens specifically in follicular lymphoma, but also maybe more subtly in diffuse large B-cell lymphoma. And I think people are beginning to wonder, and this is an extreme example. So this is an example of a patient with different tumor sites at diagnosis. An inguinal biopsy was taken, and then a mediastinal biopsy was taken. And then I think at the end of treatment, what you find here is that the inguinal biopsy, the R-CHOP removed or essentially cured that biopsy, but you continued to get resistance in the mediastinal area. And then when you get an opportunity to take these single cases, and they're just single cases, and then look genetically at these, what you can see is that you've got common genetic mutations. You've got your epigenetic mutations, KMT2D, CREB-BP. You've also got CARD11 here. But then it looks like there's been a clonal evolution, a big change. And that big change is interesting because in this example, you've got a GCB-type lymphoma in the cured DLBCL, and in the resistance, you've got an ABC type. And you can see a clear clonal evolution. And again, it shows the kind of complexities and how we need to make sure that we don't try and simplify diseases too much. And I think that people like subgroups, but you need to look like molecular subgroups and see, can you define patients into different groups? But this evolution, I think, is important. And I'll talk to you a little bit about that towards the end when we look at some of the questions. So I've only got 2 more slides. And you might say, well, where is the common progenitor cell? Where is the common progenitor cell coming into the equation here? Well, we now need to think of strategies that we can try and discriminate the CPC population from the mature tumor. And one way of doing that, and we've been very fortunate for the last few years to work with 2 Italian researchers, Marco Ladetto and Simone Ferrero, who work on MRD and follicular lymphoma as part of the Italian Clinical Trial Network. And what they've been able to do is to look at the disease, not when there's a lot of disease, but actually at the MRD stage. And they can quantify the MRD levels using the 14/18 translocation. And what we can then do is say, in the MRD stage, if a patient subsequently relapses, then MRD, essentially these 14/18 positive cells, are likely to contain both probably an enriched CPC population, but also potentially mature tumor as well. And to actually look at MRD samples as a basis for purifying, enriching for a B-cell population containing the 14/18 translocation, and by definition seeing whether that in some way gives us a better clue as to the nature of the CPC itself. And another approach, and something that that has been done and really led by a fantastic researcher at the Francis Crick in London, Dinesh Collado, is the creation of in vivo models of the CPC in the absence of tumor. And he's done that by, putting together the key mutations, BCL2, 80-90% of patients, CREB-BP, 60-70%, and KMT2D, 70 to 80% of patients. And in a way then, looking to see where, if we think now of the disease in relationship to the lymphoma tumor, but also the CPC, what we really need to do is create models where all we have is the CPC population. And then look at those models, purify those cells, get a sense of the phenotype of those cells potentially, and see then that they cross-reference with the types of mutations or the types of phenotypes that we might be seeing in our human MRD setting. And I think that this is proven very difficult to do. I think that people haven't quite as yet got a sense of what this population of cells is likely to be. But what I hope I've done today is to say, well, that's the question. You know, yes, we have a genetic landscape of the disease. We know all the mutations. We don't know how these mutations work out or play together, whether they interact with the tumor microenvironment or they're tumor specific. But I think what I believe is that we can bypass that. We can bypass evolution if we can actually target the cell that's causing the repopulating phenotype, that relapsing, remitting nature of the disease itself. So I'm going to finish by just introducing you to some outstanding questions. So I'm sold on the notion that heterogeneity is a problem. I hope you might be after the talk today, but maybe you're not. Is the CPC responsible for the incurable nature of follicular lymphoma? I believe it is, but maybe actually it's a red herring. It's just something that we've latched onto that isn't quite important. I think the question is, can we enrich, characterize, and target the CPC? And I think that that's something that Sandrine, I think it's several different groups around the follicular lymphoma community are really focusing in hard on. And I think, you know, the single cell types of studies coming through from several groups, the Vancouver group, Jessica's group in London, and also the French group is going to be key, I think, in seeing, can we tease apart what this population of cells looks like. I think we have to ask ourselves the question as well, you know, do these epi mutations, have they shared or individual effects or both? Because they're so common, so frequent, most patients could have 2 or 3 of these types of mutations. I think it's key to actually understand those. Is catalytic or non-catalytic activity more important? How do you weigh those up? Is it the tumor or is it the tumor microenvironment? And I think another question that I feel quite passionate about is that once a mutation occurs, let's say that the epigenetic mutations occur early, they could therefore be just important in initiating the disease. The question is, are they also important throughout the course of the disease? So, for example, yes, they're important in initiation. Are they important in progression? Are they important in histological transformation? And what function are they likely to be performing? Because if they don't have a function later, then if you're thinking of targeting them or working on them for a particular reason, then maybe they're not going to actually have an effect on the tumor later. They might be important in targeting that initiating population of cells or the CPC population, whereby maybe those mutations have a greater effect. And I don't think we know the answer to that, but I think it's an important question. So I've given you kind of a whistle-stop tour of follicular lymphoma, of where we've learned so much in the last 10 or 15 years, but there's so many questions that remain to be addressed. But I think one key question will be the CPC population, and I will be the first to buy the person a drink, whoever manages to find it, to characterize it, and by definition, allow everybody else to target it. So to finish, I just want to say that I've had a fantastic time in academia over the last 20, 25 years. I've been very fortunate as a scientist, as a person with a BSc, PhD, to work in a clinical environment where I've got to learn the disease. And I would urge every scientist to immerse themselves in the clinical features of the cancers that they're working on, to work in conjunction with clinical colleagues, because I think it provides you with an insight and an opportunity to think about the disease from the disease's perspective that will actually empower the research you do. in addition to typically we have a group of about 6 or 7 people. I just want to shout out to Jessica Ockerson, who is her own independent researcher now at Barts, continuing a lot of this work in lymphoma. Several different investigators around the world, and clearly the people that have funded me over a course of my career, I will be eternally grateful to them. So thank you all very much, and if anybody's any questions, I would love to have a deeper chat. Thank you so much.
Oops. Yes. Maybe I'm stuck. Okay. Maybe I can... Yes, very well. Je vais très bien. So I'm a bit freezed on the screen, but anyway, I will first thank you for your fantastic talk on common thematics that we loved, which is tracking the CPC, identifying the CPC, and eventually trying to isolate it. So I think it was really nice to have the journey of the CPC was nice, and what remains to do is quite large. So I think we are more than welcome to work all together on that topic. I just have a first question on what you mentioned at the end regarding the additive versus the separate effect of the different epigenetic modifier gene that are present in follicular lymphoma and potentially that are present also early. What is your feeling about what they can do when they are all together or eventually alone regarding the tumor microenvironment? Because we know that they have a B-cell tumor affected, but the recent studies show that they have an effect also on the reprogramming of the microenvironment. What can be really the combination of the three, let's say KMT2D, BP, and EZH2 on the tumor microenvironment? Because sometimes they have opposite effect individually, and so I'm wondering what will be your feeling on that, and what you do you have any clues? I think it's difficult.
No,
question,
I think, I think the difficulty is that we all struggle to get sufficient patient numbers. We all struggle to get the right types of biopsies to answer these questions. Whether there's a difference between diagnosis and relapse or diagnosis and transformation. And I think that in a lot of cases, people have their favorite hypotheses almost set out. And so that in a way, I think that when they're looking for function, they have an idea of where that function might lie. And so therefore there's a biasness towards the analysis that they do. So I think that the single cell analysis, when it builds up to a much larger population of cells, maybe start telling us a little bit more about the tumor microenvironment. And what I know about the single cell analysis is that it's probably much easier to tease apart changes in the tumor microenvironment cells than it is looking specifically at the tumor B cells. But I think that what the epigenetic mutations are doing, I still think is an open question, whether they're having a multitude of small effects, or whether they're altering one particular pathway or two particular pathways that are actually impacting on the tumor microenvironment. And I go back to the kind of question as well as the timing. Are those epigenetic mutations having the same effect when they occur in a pre-malignant, in the initiating of the disease? And do they retrain the local microenvironment there within the germinal center? And is that the same then subsequently when there's a malignant phenotype? And they're not easy questions to answer, but I don't think people have said, this is really difficult, and these are all the problems that you can see if you're only looking at 4 or 5 different biopsies, and you feel lucky to get data on 4 or 5 different biopsies. But does that capture what potentially could be happening when the mutation component could be slightly different across the different biopsies that have been looked at? Yeah, I'm confused. I don't have an answer. I can ask the question, but I don't really have an answer. And I don't think the data that I'm seeing, Sandrine, maybe you see it differently, or Oliver, you see it differently. But
no, I think
people are
I don't
trying,
have
but they're not answering the question.
No, but I think it's a difficult question. It's just we try to model that in mice, but it's super difficult because we know that the kinetics can be important, and we don't really know who is first, who is second, who is the third mutation, and maybe the kinetics can make the interaction different as well. And it's quite difficult to address this aspect from a B cell point of view and also from the tumor microenvironment. So I don't have any cues on it,
Yeah.
but it was just to have your feeling.
Yeah.
I think we have question maybe. Do you see it?
So maybe I get
Question.
started with Laura's question.
Yes.
And I think I had more or less the same question, Jude. Excellent talk. Thanks so much. My simple question would have been, what do you think is the first hit in lymphoma genesis? Is it the 14/18 translocation? Or what I think Laura is also implying in her question, can it also be CREPP-P and KMT2D mutations, any other epigenetic mutations? So where do you think, at what stage do you think CREPP-P and KMT2D mutations occur?
Well, it's like I've always felt that these mutations occur in the germinal center. So like you got your 14/18 clearly doesn't, but all the other mutations are probably biased towards the germinal center. That's not to say that, there's patients with a syndrome called Rubinstein-Tybee syndrome that have germline CREB-BP mutations, and there's some evidence to suggest that some of those patients go on to develop follicular lymphoma. So clearly, in certain circumstances, you don't necessarily have to have a germinal center phenotype, and I think Brian Huntley has shown some evidence in relationship to these mutations occurring pre-germinal center. But I personally feel that if you're looking in the round, the majority of these mutations must be occurring as part of the somatic hypermutational process or within the germinal center. But it's also interesting to think that if you look at pancreatic cancer, if you look at a lot of different solid cancers, these patients have mutations in CREB-BP, have mutations in KMT2D. So it doesn't necessarily have to be germinal center derived. But simplicity for me, when I think of the CPC, it has experienced the germinal center, it has undergone somatic hypermutation, it has acquired mutations, but there are going to be exceptions to that rule.
There was actually a question from Bertrand Ardel. I can no longer see it in my chat, but I saw that Bertrand was also online. Bertrand, can you speak up?
Yes.
Or can we maybe see the question one more time?
Oh.
Okay.
So maybe in the meantime, I have another question, Jude, to fill that gap, if I may.
Yes, go for it.
So conceptually, Jude, is the CPC not yet fully transformed, or can that CPC actually fulfill all the criteria of a fully transformed cell?
Well, I think because the first single cell data was done by the Marseille group, and Pierre Melpied, I think, was the lead author. What he showed very nicely is the continuum of cellular diversity within the germinal center, from the dark to the light zone, and then potentially cells moving back and forth. So I think that you have to think of the CPC is probably a continuum of cells that fall within a certain remit. There can be, you could say, there could be mature and immature CPCs of sorts. But so I think that we have to look at that population not as a single population, but having a potentiality to have a dormancy, or to be able to evade treatment for some reason, and then to reawaken if it acquires another mutation, or the tumor microenvironment might change. And so that when you talk to patients with follicular lymphoma, sometimes they tell you that, this happened to me before, I had an infection before follicular lymphoma came back. You have to think about the complexities that could actually be causing reawakening of the cells. But I think, again, it's an open question, but I think hopefully some single cell analysis or some way of enriching for the CPC will empower us to get a better sense of what those cells are actually doing. And maybe it could turn out that the CPC doesn't exist because it's all based a lot of the time on genetic data, not necessarily phenotypic data, and we don't have any self-surface notion of is there something special about that population of cells.
That was actually, do you hear me? I got the privilege to be able to join you. So thank you very much, Jude. That was a fantastic talk. We agree on everything, so I don't have many questions, or we have the same questions. But that was, to follow up on what you just said, I think we have to realize that one of the big difficulty in fishing CPC is that we're going to have to fish many flavors of CPCs, many type of maturity in their development from very early to very late, and they of course will appear differently depending on the treatment as well. And that was my question. Do you think that we're gonna have always the same, let's say 2-3 categories of very immature precursor cells that will have pretty much the same alterations, or that the CPCs can be a very complex cell that will appear differently with different type of therapies?
No, and I think the difficulty is that there's probably many ways to deregulate a gene. So you can imagine that specific mutations are clearly there, but you can see examples where a gene could be silenced. The expression of a gene might be downregulated to sufficiently so that it kind of phenocopies haploinsufficiency or loss of function in some way. And it only has to happen in a population of cells, in one cell in the population. And then that cell, if it has a 14-18 translocation or it has other potentiality, can actually progress. And what we don't know is whether these individual phenotypes are maintained in throughout the course of the evolution of the disease. So I think, Bertrand, you know, when we go back, we were, and Sandrine is the same, and I think, and Laura, who's online, is the same. We were young people when, and the same questions are still there, except that we've got a much greater sense of the disease itself. We've got a much better sense of the genes, the genetics, and the complexities. But are we any closer to identifying the CPC itself? I don't see the data yet, but I think everybody's trying and everybody's looking at it in a slightly different way, and I think the hope is that you know, if you can sequence enough single cells and you've got enough bioinformatics people to tease apart and cross-reference with potentially mouse models, etc., you might get a flavor of what that population is like. The worst thing is, Bertrand, you might be looking at it and you just haven't recognized yet in the single cell work that everybody is doing that we actually have it and it's there, but we don't quite know, or we haven't the right binoculars on to see it. But it seems a long time since we met in North Berwick, probably 15 to 20 years ago when your first papers were coming out. And these are the kind of questions that were there then, and maybe we've got more granularity on the questions, but that key question still remains.
I was afraid you were about to say we were young then and we have gray hair now, and still.
Well, you have gray hair. I don't have any hair, so you know things have changed indeed.
So, Jude, which compartment do we have to sample? Let's say by single cell to identify the CPC. Is it the bone marrow? Is it tissue, lymph nodes? Where do we have to look for it?
So I think that in our case, and I think we're all working on a budget. So you know, in research, you're working on a budget, and so I would have theoretically reasoned, you know, bone marrow would be great, bone marrow in remission, bone marrow where patients have been on maintenance rituximab, and then if you've collected those samples and those patients have subsequently relapsed, then any B cells that are present, or any 14-18 positive B cells in that compartment, are likely to be potentially CPC-like. What you don't know is how dynamic those cells can be, how much changes can actually they undergo. But if you were writing for an exam, that's probably what you'd think. That's the population that would give you an enrichment. But then you need to cross-reference that with the single-cell data, with your mouse model data, and see whether you can build a case for that cell population to be important.
But Jude, don't you think that part of the answer is also what is triggering the BCR of those CPCs, and it can be many ways by specific antigens in some case, or maybe early on, and then getting independent from that later on, and maybe some environments such as the bone marrow are more in this later phase of that, because the microenvironment adapted to be a stimulator of those CPCs, and that there are several phases like that, and depending on the nature of the first stimulation of the cell, and you might have CPCs in the liver or CPC somewhere else in lymph nodes or in bone marrow, but depending on that immunological factor as well.
Yeah, and I think that because there's now a focus on monitoring disease over time, you've got an opportunity potentially with taking sequential peripheral blood and bone marrow samples to ask the question, is the microenvironment changing? And does that change in the microenvironment then predate the expansion of the cells or imminent relapse? So like the question that you said, I think we need to we need to knock on multiple different doors, and hopefully one will open sufficiently that will give us a clue that the NK cell population is increasing by twofold, and that in some way then is driving a phenotype or driving an immune change that's actually allowing for the CPC population to reawaken. But I haven't seen data to suggest that those things are I think the experiments have to be completed. They have to be completed in a fairly large number of patients, and those patients have to be homogeneous in some fashion in relationship to the genetics to see, can you tease apart probably not one, but a number of different mechanisms that are causing, as you said, Bertrand, this reawakening of this progenitor population of cells. You can feel a bit despondent because it is difficult, and I think that we've all worked very hard in the last several years to try and get close to the answer, and I think we may be within touching distance, but it's like winning and losing. It's very binary, and currently I think the journey is proving far more challenging than I think you got the notion in solid cancers as well about these persister cells, or the fact that we now begin to realize genetic changes aren't everything, and then that the rewiring that you might see due to epigenetics and changes in gene expression and subtle changes could be important in drug resistance, etc. It all feeds together into the same type of pot, and makes it very challenging indeed. But we've come a long way, and I think we're asking the right questions. And I know from the community that there's a lot of clever people working very hard on trying to get the answers.
I think we have a question, an additional question from Laura. Follow-up question. This one. Also, assuming that BP and KMT2D occur early and are not sufficient to drive malignant transformation, what do you think determines the evolution towards FL versus EZB and C3 DLBCL?
But it's like when we're all asked, if we're asked to write a paper on histological transformation, I say, well, I don't know what to write anymore, because I don't think there's anything that I know is a driver. And people talk about OP53 and MYC and CDKN2A and all these things, but I'm looking for something much stronger, something that I can say 60% of the aggressive disease is due to this particular mutation, that particular mutation. And so I don't know the answer. And I suppose you either believe we have the answer or you believe that there is a better answer out there that kind of encompasses the aggressive disease. And then you have the notion, as I was saying, Laura, about the new data. Not only do you get DCB transformation, but there's ABC and there's non-mediastinal. And so in a way, and then you've got the notion is the DLBCL, is there something different about that if it occurs a first relapse or occurs later on in the disease? And I know I'm kind of fudging an answer, but the reality is I don't think there's an answer, and I don't think the data is there telling us what exactly is driving the aggressiveness of follicular lymphoma. And like it could be, Jude hit is there for sure, but I'm looking for something more credible and maybe different. But maybe the answer is there. And, by all means, come online and tell me I'm wrong, or tell me that the answer actually is no. But don't. There's no worries either way. But I think the key thing is the questions, and what you can see is that donor-derived bone marrow case and Oliver found one in the Dana-Farber as well. These clinical examples change how you think about things. I'm sure it's the same, Sandrine, for you when you started to look at the evolution in the EPIC study, that you begin to home in on what you think is the key question.
Actually, Jude, Laura cannot have the privilege like me to join live, but she's asking. I ask you because I am not sure EZB, DLBCL de novo is a transformed FL since they behave so differently.
And I think Laura's right. I think that there has been a notion that maybe they actually are more similar than other types, and that you're getting you never picked up the FL, and essentially at diagnosis then you've just picked up the DLBCL component. And that's why in some cases there's a lot of data looking longitudinally at patients with diffuse large B-cell lymphoma and some relapse with FL. And you can see in those cases that suggest that there was a preceding FL before the de novo diffuse large B-cell lymphoma arose.
Yes.
So Jude, thanks again. As so often, I think really good research yields even more good questions. So we're left with a bunch of really good questions. So tell me, like at AstraZeneca, will this company allow you to keep working on that? Can you still try to tackle CPCs?
I think that the new role is something called a VP in Haematology Discovery, and so that in a way, the drive will be to try and discover new biology that can be important in understanding these diseases and then doing better for the patients. And I think that we plan to, and we are collaborating with the best researchers around the world across a lot of different hematological malignancies to try and see can we do this better than we have been doing. And so in a way, I'm very much interested in the research that's coming out at the present time. Looking forward to meeting everybody at ASH next week, and fingers crossed in relationship to follicular lymphoma. Although I've left academia, I'm really excited to follow the research that's ongoing, and whether we can cure the incurable at some stage very soon.
Yes. Maybe I just have a question regarding the definition of what we call a CPC, because as you have seen on your scheme, we have the one we catch with the EPIC cohort are the, let's say, early CPC, so before the disease happened, and what you are proposing in the follow-up of the MRD is another potentially other kind of CPC. Do you think they are similar, or do you think the treatment imposes some plasticity to those cells, or do you have any, or do you think they are the same and maybe they are just in a niche and they are just quiescent and they just reappear? Do you have You see, I have the same. I have this question a long time. I don't have answer.
Yeah, no, and I think it's a very good question. That depending on where you look and when you look, you're going to find a slightly different population of cells, especially because at that stage, they won't be treatment naive patients anymore, and so therefore there's you just don't know what treatment is actually affecting that particular population of cells. I think we need to go back and when we found the answer, we can go back and answer those questions and say to ourselves, well, yes, they are very similar, or they've changed somehow, or certain mutations changed the phenotype of the CPC in a much stronger way than others. But I think great questions, and it also shows the complexity and the difficulty in identifying populations, interpreting data is going to be very hard, I think, to effectively identify a population, and then how you go about testing whether that's the population that's driving disease relapse.
Yes, and I think it's really hard to get pre-diagnostic samples, particularly in follicular lymphoma. It's not like MGUS or MBL or whatever. It's just not the same, and so it might be difficult to go beyond genetics.
And I think you have to do, but this is why I think the minimal disease setting at least might give you a chance, or you sequence at the single cell level so many cells that you feel you've got a depth that you can capture a CPC if it's present within the germ center population.
Jude, another question related to your idea of cell-to-cell epigenetic heterogeneity, which is, I really am a strong believer of that. Do you think this is heterogeneity driven by genetics, or can this also be functional heterogeneity, given the fact that epigenetic marks and interaction can be reversible at some stage?
First of all, I can't take credit for that. I read it in a Trends in biology. I really thought the figure was very, very nice indeed, and it seems intuitively correct that you're not going to lay down these marks so perfectly from a cell-to-cell perspective that you're going to get subtle changes in gene expression. As I said the tools, say for example, that we use to measure gene expression. I use an example whereby if we all got a 20% increase in our salary, it would make a measurable difference to us from a life perspective. But it's very hard to measure subtle changes like 10% or 15% or 20% in gene expression. But if these marks are different from a cell-to-cell point of view, then certain genes could have subtle normal dynamic differences in gene expression. The question is, as you said, is whether it's a general effect on gene expression, or is it specific to certain genes? And I think maybe the tools will be coming soon, whereby you can ask those questions. But currently, I think it's another good question that sounds good, theoretically makes sense, but actually needs to be proven. I think everybody should go for their evening dinner in
So we definitely have to keep discussing that at ASH. I hope we'll be meeting at ASH, Jude, Sandrine, Bertrand, hopefully many of you in the audience.
With great pleasure.
Yes, we'll be there.
Again, this has been exciting. As I said earlier, I'm a first-time participant, but I'm a fan already. So I will try to join in many more times. This is an excellent session. Thanks again for inviting me. And Jude, this was outstanding.
All the very best, everybody. And it's just great to see so many friends and everybody online. Thank you very much for your attendance. And thank you very much, Bertrand and team, for the invitation.
Thank you very much.
Yes, thank you very much.
Have a nice evening. Bye.
Yes.